All right, we're back. Now we're going to talk about the glucose storage device in animals. So starch is how plants can store glucose. Glycogen is how animals or we can store glucose, right? So glycogen makes up a large portion, makes up 10% of our liver mass and a small amount of our muscle mass. It's how we store energy. Okay, it looks very similar in shape to starch, except for it's even more branched. So what do I mean? We're going to go back and we're going to look at the structure of starch. So going back. All right, see the structure of starch? See that the only way that starch branches is it's still 1,4 branching. It just branches down, right? Well, it does this sort of extra linkage down and you get this new chain, but it's a flat chain that links down to another flat chain that can link down to another flat chain, right? That's what amylopectin looks like. That's how starch can link. But let's look at how glycogen links. So glycogen can do this. So what you see is you see that one unit of glucose can link to 2 other units of glucose at the same time. That's not what we see in plants. Okay, so what does this mean? This means that we can access more glucose and release more glucose more readily in animals than we can in plants. Okay, so that's the difference. You still have your 1,4 linkages, but you could also have a 1,6 linkage. Okay, this is your 1,6 linkage, but you still have your 1,4 linkages. So more linkages equal sort of more branching. So you got more branching, so shorter amounts of residues in your branching, and that means that you can release more glucose at once. Or what's interesting here is depending on how you sort of hydrolysize this, if you hydrolysize it with alpha amylase, you get glucose, but if you hydrolysize it with beta amylase, you can actually get maltose. So we can get 2 different types of sugars in animals as well. And how you can identify if you have any glycogen present is you can run the exact same test that you ran on starch, except for it's not going to turn blue, it's going to turn red-violet. Okay, so a different color than what you saw in plants. So a little bit, another color test, which is nice. Color tests are easy to do, right? Those are preferred. All right, and then we have our group that I like to think of this as sort of like the periodic table has metals and non-metals, and then you have the metalloids, which are like kind of the weirdos. Well, for our storages, we have the major animal storage, glycogens, and we have the major plant storage, starch, and then we have this other category that can exist, and it's kind of an interesting category in the sense that it has this unique 1,6 branching, but it can kind of do whatever it wants. It can branch at the 2 position, 3 position, 4 position. It can basically do whatever it wants. It's formed by bacteria, and so some places that you could find it are in dental plaque. So this is sort of like the category of the kind of randoms, the ones that don't fit the major categories that we previously talked about, right? And those are it. Those are your storage polysaccharides. Okay, so your storage polysaccharides are, you got starch in plants, you got glycogens in animals, and then you got dextrans. Okay, those are it. Those are the ones that we're talking about here. All right, so what about your structural polysaccharides? So your structural polysaccharides, these, interestingly enough, they don't look very different than your storage polysaccharides. And that's probably not that surprising because we're not dealing with a bunch of different residues, right? There aren't that many residues that are going to appear in a polysaccharide, so they are going to look all very similar, right? And the most abundant structural polysaccharide on the planet is a natural one, cellulose, right? It's found in the cell walls of basically all plants. It's why trees and plants have their strength and support. We cut down trees and we pull the cellulose out and we use it to make paper, right? Printer paper, or depending upon how you prepare it, maybe some good old super duper valuable right now, TP, right? That toilet paper shortage, whoo. Never could have imagined there being a shortage of toilet paper, but that is the world we live in now, right? and we need cellulose for that toilet paper. My brother, random side story, used to work for a Georgia Pacific mill, which is a cellulose plant out in Alabama. And as an employee at the cellulose plant, like he would get bonuses, and his bonuses would be he could take like a box of paper products home. So there was a solid year there where I never had to buy paper towels or toilet paper because my brother would get like boxes of it like once a week, and he wasn't going to use this huge crate of toilet paper. So we shared it with us all. If only, right? If only that was now. But nonetheless, let's look at what cellulose looks like, right? Okay, so this is cellulose. It is very similar to what we saw with glycogen and starch in the fact that it is a 1,4 glycosidic bond, but the difference here is it is not an alpha glycosidic bond, it's a beta glycosidic bond, so it's going up, right? Now, you may look here though and say, well, wait a minute, now it's going down. Oops, sorry. Now it's going down. It must be an alpha, right? But take a look again. Look what happened to the structures. This structure has been flipped upside down, right? So this looks like an alpha, but it's a beta. These are all beta linkages because basically hold your hand out in front of you flat with the ground. So that's one. Now flip your hand over 180 degrees. That's the next one. Now flip it back and then flip back, right? That's what it does. Just imagine your hand flipping up and down and then linkages of hands. That's basically what's happening here. And you end up getting what looks very similar to like a beta pleated sheet kind of scenario. And so here is an example of what that looks like. And just like, remember how you had these covalently bonded beta pleated sheets that could hydrogen bond with another beta pleated sheet? Hey, guess what? Same thing, right? Same thing that's happening here. So here is your one chain of cellulose, and it can hydrogen bond with itself, right? Or it can choose to hydrogen bond with another sheet directly beside it. But here's the difference. Here is how the beta pleated sheet differs, okay? Because proteins don't do this, but we see this with the polysaccharides. With polysaccharides, now imagine that not only do you have a whole bunch of chains in the same plane as one another, hydrogen bonded to each other, now you have chains above them, and they can hydrogen bond with those. And you have chains below them, and they can hydrogen bond with those. Now you really get to see what's happening here. This is where you get all the structural strength from. But can you tear paper? Yes. Can you tear toilet paper? Yes, it's even easier, right? Because what you're doing is, yes, you get strength, but the strength is hydrogen bonds. It's not like when you tear a piece of paper, you're tearing it sheet to sheet, right? So it's not really that hard. Or have you ever tried to tear something before and it seems really hard, but then you slide your hand over or come at it from a different angle and all of a sudden it's a lot easier? Maybe some of this has to play with that, right? What were you trying to break before? Okay. All right. So what are some other structural polysaccharides? Well, chitin is a very common one. It's what makes up the exoskeletons of different crustaceans and stuff. So I played this video game on PS4 called Ark Survival, and it's kind of cool and interesting. You're like, you wake up on this weird planet with this device in your hand, whatever, yada yada. But there's dinosaurs and there's all different types of animals from sort of that era. And you need chitin. That's one of the resources that you need. So you have to kill these little crustacean creatures so that you get the chitin from their exoskeleton. You can use it to make armor and stuff, right? Why? Because, and it's hard to get, right? Because chitin is like one of the strongest armors you can have in the game because chitin is so strong. And yet it does not differ very much at all in structure from cellulose. I would not want to make an armor out of toilet paper, right? I mean, maybe you made yourself a mummy one time when you were a kid out of toilet paper, but you wouldn't want to go into war with that, right? So what's the difference? I mean, the difference is this. It's not very much. So the only difference is instead of a hydroxyl at this C2 position, you have an N-acetyl group. And that's really it. And that makes all the difference in the world as far as strength is concerned. All right. So, but let's talk about all the different forms that we can have, though, right? So, natural cellulose, right? Just like how we could have parallel beta sheets in proteins or anti-parallel beta sheets. Guess what? Same thing here. All right. So, since chitin is just like cellulose, we can have parallel or anti-parallel chitin, right? But there's actually three different forms of chitin that you can find. So alpha chitin means that it's all parallel arrangement of chains. There's also something called beta chitin, which is all anti-parallel arrangement of chains. Okay, so then what is this last form of chitin? Well, this last form of chitin is gamma chitin, and what gamma chitin is it's basically it has both, right? So you've got some parallel sheets that are then separated by a singular anti-parallel sheet. There's not really a picture of this in your book. So what I'm going to do now is I'm going to jump out of this PowerPoint and I'm going to go and I'm going to show you a picture. All right, so here's the picture. So here's alpha. Well, it is constantly sort of anti-parallel, right? And then your beta chitin, which is purely parallel, okay? And then here's your gamma chitin, where you've got two parallels broken up by a singular antiparallel. And then two parallels broken up by a singular antiparallel. And these are basically your three forms that we see in nature of the chitin, right? And so this sort of in acetyl group, this is your reducing terminus in chitin. All right, so let's go back. Okay. All right, so now moving forward. So that's chitin. Let's look at agarose. So agarose is an important polysaccharide mixture that's isolated from this marine red algae that's called agar. And it basically consists, so what agar consists of, it consists of two components. So the major one is agarose. And what agarose is it's this alternating, it's a chain of alternating galactose and then this sort of anhydrogalactose that has some side chains of a methyl galactose. So what this is these are basically just sort of forms of galactose that have been modified, right? But you see that this does form a double helix corkscrew type structure, okay? But it's large enough though, this cavity through the center is actually large enough that water can actually fit inside of it, okay? Which is actually really cool. That water can not just be on the outside, water can actually fit inside of this helix, because most of the reason that you would form a helix would be to avoid water, but this is actually a very like big helix, so water can actually go inside. All right. Another thing we need to talk about are these glycosaminoglycans. Okay. And these glycosaminoglycans, these are repeating disaccharides in which one of the monosaccharides is an amino sugar and one of the units has a negative charge associated with it. That's either a sulfate or a carboxylate. One of them has to. It may be both. So in this case, here's your amino one, and they actually both have negative charges. Or if we look at this example, again, amino, they both have negative charges. This is your amino, only this one has the negative charge, right? So they don't both have to have negative charges, or in this case, the amino one doesn't have the negative charge, but the other one does. So you kind of have options here, but this is another structure that we see commonly formed by these polysaccharides. And this isn't really a polysaccharide, this is a, so the these have these repeating disaccharides within them. Okay, so in the next video, we're going to talk more about what these glycosaminoglycans are and what these different forms are. All right, so I'll see you in the next video.